Good morning, everybody. Sorry, puppy issues. All right. So, whew, chasing puppies around and stuff like that. It's not for the faint of heart. Anyway, where was I? Okay, so what we have to do today is to finish off translation. And let's bring up this. Yada, yada, doo. All right, so as far as I can remember, we finished off at the initiation. Let me make sure. Yeah, I'm not muted or anything. Initiation of translation. Oh, got my coffee this morning. Fabulous. And I think we've done a pretty good job of covering that. If you have any questions, feel free to chime in. But if not, I want to show you a few slides of an elongation video. So this is linked in here. And let me just, which one is it? That one, I think. Oh, fudge nuts. No, not that. Gonna go back again. Sorry, the bar that controls Zoom hides all my tabs, so I have to be really careful. There you go. This is the one. Okay, so this is the elongation video linked in the PowerPoint, and I'm just going to put it in the chat as well. Pardon me. And one of the things that you need to kind of be aware of, right, which is kind of ties into like the big scale organismal sense, is that translation is very energy intensive. So for one amino acid, you need basically 2 GTP, right, which is kind of analogous to ATP, and 1 ATP. Question for the class. Can anybody remember, we haven't got to the GTP stuff yet, or we kind of have, but not super well. But can anybody remember where 1 ATP is used in the addition of an amino acid to a polypeptide? Think about the whole process. This would make an excellent exam question, which is on Thursday, don't forget. All right, so where is 1 ATP, 1, used in the process of adding 1 amino acid to a polypeptide chain in translation? Kind of a difficult question, but I'll walk you through it as well. Okay, so what's the because I'm getting nothing but silence here. I can't even see Tanya to see whether she's frowning at me or not. What's the very first step? What's the first thing that you need to be able to add an amino acid? No, not quite. Good thinking though. Phosphodiester bonds are in the actual mRNA themselves, so not. I mean, obviously you need that, or even the energy in the phosphate bonds, the phosphate to phosphate bonds. Yeah, that's where the energy is coming from. But what's that energy used to do in translation? What's the very first step, really, if you think about it, in the addition of an amino acid to a polypeptide chain? What's the first thing? I'm not talking about translation, like the initiation and translation, stuff like that. We're talking about what, like if we find an amino acid, you know, floating around in the cell, that one, right? How does that get incorporated into that protein? Going to get you to think a little outside the box. How does that amino acid get added to that protein? What does the translating part? Also has a T at the beginning of its name. Just one T. Staring at you on the screen. There's one already attached in the ribosome. Squiggly thing. What does the translating between the 3 letters of the mRNA sequence, the codon, to an amino acid? What's the intermediate? Come on, peeps. tRNA. Yay! Awesome, right? Begins with a T. Stands for transfer, but translating RNA would do as well, right? Okay, so what's that tRNA carrying? Stuck on the top of it. Yeah, it's carrying an amino acid, a very specific amino acid, right? In this case, it's methionine. Could be any depending on the tRNA. How does that amino acid get added to that tRNA? Is it just random stochastic chance? Or is there something that does that with a high degree of specificity? What adds the amino acid to the tRNA? We covered it like a couple of slides up. Yeah, the amino acyl synthetase, right? What does the amino acyl synthetase use in form of energy to do that? ATP. So before you even begin translation, you have to charge your tRNAs, otherwise, they're not going to bring any amino acids into the ribosome. It takes energy to do that, right? So that's 1 ATP alone required for adding 1 amino acid, and we'll see where the GTP comes in. Okay, cool. So good stuff. So really what I want to, part of the reason for kind of going through that kind of roundabout way of explaining things is that I want you to think about this not just in a series of dang, 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 facts, right? But think about it in the kind of the whole context, right? Where's energy used? What are all the different things involved, the players involved in this process, right? Because they're all required, otherwise it doesn't work. So I'm just going to turn the fan on so that I'm not. Starting to get warmer. All right, so essentially what we have here is the initiation of translation has already occurred in prokaryotes. It's prokaryotes. Why? Well, because it's in the title of the video, somewhat unhelpfully, but also because there's a Shine-Dalgarno sequence here too, right? And so that tells you it's prokaryotes because in eukaryotes, where does the small subunit of the ribosome bind? Come on, exam on Thursday, right? You gotta like know this stuff. What does a small subunit of the eukaryotic ribosome assemble around or bind to in the initiation of translation?
Wasn't it like a bunch of like the F something?
Yeah, that's very much true. Where are those EF somethings located on the mRNA? Are they located at the start codon? Going to keep wagging my
Oh,
eyebrow.
because on the prokaryotes it starts when it initiates, right? And the other one, it's like it's not there at the location of the starting point.
Correct. That is completely correct, right? So it's a scanning process. Where does it scan from to get to the start codon? Where does the thing assemble before it kind of processes along and finds the start codon? Super important in mRNA stability too, gets added during RNA processing. what are the two things that get added to the ends of mRNA during mRNA processing
The
in
5
eukaryotes?
caps,
5' cap and the
polytail.
poly A tail, right? So both of those are important in the assembly of the small subunit, right? So I could show you a picture of this if I was so inclined and ask you, is this eukaryote or prokaryote? And you'd be able to say, without looking at the title, it's prokaryote, right? Because it has the Shine-Dalgarno sequence, which is involved in guiding the small subunit to the start codon. Okay, cool. So hopefully these kind of guided questions help, right? Because I want you to figure out what you don't know before the exam. I don't want you to figure out in the exam. I'm not just being a jerk, honest. Okay, so, right, back to the point. We have our full ribosome assembled. We have our small subunit down here, which binds to the mRNA. We have our large subunit up here, which have these three sites: access, peptidyl transferase, or peptide bond forming, and our exit site. And the tRNA with methionine in both prokaryotes and eukaryotes is assembled in the P site, right? And this kind of tear-shaped blob up here is the peptidyl transferase, right? And that's why it's in that particular place. And so what we need to do now is we need to bring in another tRNA, whatever is carries an amino acid for the UCC codon, right? That's going to come in, and then we need to do two things. So essentially there's three steps in this. tRNA comes in, this amino acid gets cut off of this tRNA, and then a peptide bond is formed between this amino acid and the one that is coming in. And then, I guess there's four steps actually. Then this whole thing processes along, right? So essentially, I'm just going to write one for the sake of time. Hang on, charged, not traged, charged tRNA enters the A site. Amino acid attached to the tRNA in the P site gets removed. Peptide, I'm writing this in the chat box just so that you have a record of this, is formed between the removed acid and the, I hate writing out amino acids, I always just put AAs on the new tRNA in the A site. And then the final step, ribosome processes along or moves, right, if you want to use a different word, the mRNA by one codon. That's essentially what elongation is. Those four different steps. Come on, up you get. Don't know why that did that. So essentially, and there are some other things involved. So there's a couple of proteins involved, and there's going to be two GTP used. Right, the first one, trundle, is to bring in, oh no, stop it. To bring in the new tRNA, right? So UCC encodes serine. Let's see if we can find that on here. So I'll move stuff out of the way so I can see what I'm looking at. So UCC, there you go. Actually, UC anything encodes serine because we have that degeneracy at the third base. Right, so UCC is serine, easy peasy. Protein called EFTU, right, in prokaryotes, and I think it's EF1 in eukaryotes. Let me see. Come on. Here we go. EF1A. There you go. Is involved in bringing this tRNA in, right? So these tRNAs just don't kind of float in, you know, of their own accord. They're actually actively guided. Oh, hello, Tanya. Guided in by this EFTU with the help of GTP, right? And so once this comes in, that GTP is going to be hydrolyzed. Bring it along a little bit. You can watch this video yourself as well. It's got a good kind of narrative that explains what's happening. Right, and so now that comes in, this GTP is going to be hydrolyzed and the EFTU will be removed. Where are we at? Come on, out of it. Oh, there you go. Right, so that's gone. That GDP, right, so it's exactly the same as ATP to ADP, right, to generate that, or to release that energy rather. That GDP is then swapped out for another GTP later by another protein, which you don't need to worry about. Okay, so now we have two tRNAs, one in the P site, one in, which was there originally, one in the A site. Now there's a enzyme, well, it's actually a ribozyme, right? Anybody remember what a ribozyme is? What's it made of? What individual, what sort of macromolecule is a ribozyme? Quit it. Sorry, keep clicking the wrong thing. What's a ribozyme? Keys in the R. It's an enzyme, but is it made of protein? What's the thing that does the catalysis in the ribosome? Is it protein or is it? It's not DNA. What's the other bloody nucleic acid that we're talking about? Begins with an R.
Ribose. So it's a sugar.
Nope. Well, it has sugars in it. Almost. That's sorry. I actually forgot that was another thing that began with an R. Sorry. Small R and then a big R. RNA. Right. Oh, I'm sorry, Levia. I didn't see that in the chat. I was just focused on like goading Tanya into giving me the answer. Yeah, it's ribosomal RNA. Right. Oh yeah, and Kim, thank you for the support for Levier there. So it's ribosomal RNA. And that's a ribozyme, right? Ribozyme. Hello, Lilybit. Go on, up you get. Up. Come on, up. There we go. Sorry, got a little canine distraction here. Where is she? There you go. You can also see my foot. If I jump and go, ow, it's because she's bitten my feet. I don't know why she does that. Perhaps they taste nice. Right, so it's the rRNA that does the work of forming this peptide bond. And that's really important to go over because it's the protein kind of gives the context. The shape holds things together in the right way. It's the ribosomal RNA that is the peptidyl transferase, this kind of tear-shaped blob here. And so its job is to cut off this amino acid. Let's see where. Okay, it's highlighted in yellow there. Come on. Not quite. Right, so it removes that and then it catalyzes the formation of the peptide bond between the carboxyl group on this amino acid and the amine group on the one that is still attached to this serine tRNA. Let's see if we got. Come on. Here we go. And so you can kind of see that in kind of more detail here, essentially. Right. So this, don't worry about this thing in pink. Essentially, we're going to be doing a dehydration reaction between this carboxyl group here, not the double bond. It's actually this one down here, and the amine group in the new amino acid. So you can see that just there. Can you give me a second? I've got to let the little pest out so that she can be with the other dog. I'm back in just a second. All right, sorry about that. Got the other dog in here instead. Okay, so now that we have that peptide bond formed, we now have a dipeptide, right? We have the first amino acid, we have the second amino acid. These are now peptides because they're part of a chain, right? It's just same thing, kind of different context, essentially. Okay, but now we can't add another one, right? Because our A site is blocked because we have this serine tRNA with these two peptides attached to it. So the whole thing moves. The tRNAs stay put, essentially, right? They stay attached to or hydrogen bonded to the mRNA. But with the aid of another protein and another GTP, so this is the second GTP used, this whole thing processes along one codon, exactly. Right, and so we've got another protein, this is EFG, which is EF2, I think, in eukaryotes. That now binds to the small subunit. The GTP gets hydrolyzed, right, again, releasing that energy. And it's just caught in the middle here. All right, a little bit further. The whole thing shifts along one word. This is why where that AUG, that start codon is, that's what sets the frame. So the words are 3 bases after that. Every 3 bases in the mRNA after that first word are the subsequent words, essentially. Right? That's why the AUG is so important. It's also why the yellow mutation that we're studying in lab results in no yellow protein being formed because that G is changed to a C. It's a point mutation. So no start codon, no protein, right? Okay, now the ribosome's processed along. Now our A site is open. We have our polypeptide or growing polypeptide chain attached to the amino acid in the P site, just as methionine was before, if you remember. And now we have a discharged or an uncharged, I'm not sure, correct word, tRNA in the E site. This will now leave, gets kicked out, oof, done your job, and it will be recharged by amino acyl synthetase, right? So it can start the whole process again. Now we have the same, essentially we've reset the cycle. It's almost like PCR, right? Now we go back to the first step. tRNA comes in, which I think they've got. right? This one is, oh, thank you, YouTube. This one's for lysine, AAG. Okay, brought in with our elongation factor, that's why it's an E. IF is initiation factor, EF is elongation factor. That GTP is going to get hydrolyzed, so this leaves. And now the same steps occur again. These two amino acids are picked off, right? So the bond here between the serine and the tRNA is broken. Peptide bond formed between serine and lysine. Whole thing moves along with the aid of another GTP. Another amino acid comes in, and the process continues until we get to the stop codon. Right, and so you can see that there, essentially. It's just a series of repeating through those 4 different steps. So 1 ATP to add the amino acid to the tRNA, 1 GTP to bring the tRNA into the ribosome, and the second GTP to move the ribosome 1 word along, 1 codon along. So essentially, for every amino acid that's added to that polypeptide chain, takes 3 units of energy. So you can see when organisms are under conditions of nutrient limitation, or as we call it in humans, starvation, protein synthesis is one of the things that gets cut almost immediately because it takes so much energy to do so. That energy could be better used potentially to just maintaining the function of the organism rather than growing or producing new stuff. Anybody got questions about any of this stuff? It's kind of one of those complex things that's actually quite simple once you get it. It's just those 4 different steps occurring over and over and over again. And proteins can vary from being real short. Like one I knew someone working on was like 8 amino acids long, barely even called a protein, to be honest. That's oh shoot, I forgot what the name of it is. It was a reproductive hormone. Can't remember what it was called. Gonadotropin releasing hormone, I think, maybe. I don't know. They're going to try and use it to sterilize possums. Ha, that didn't work. Not opossums, those ugly, nasty-looking things that you have here in North America, but possums. Possum? Anyway, that's a whole nother kettle of fish. Anyway, they can range from really, really short to being monsters, like single polypeptide chains of, up to 3,000 amino acids long. So this has to happen over and over and over and over again for every single one of those amino acids. Okay, how you all doing? Wishing it was Friday already? Tell you, this semester feels like one long slog. I really do miss our spring break. Really could have done with that. Anyway, not necessarily to go like partying down the beach and stuff like that, but just having a break. So, and I'm sure you will feel the same too. Boop-a-doop-a-doo. Okay, so essentially this happens continuously, right, until we get to a stop codon. And stop codons are codons that do not have a matching tRNA. So if you look at this codon table, and this looks kind of like wildly confusing, but it's actually fairly simple. This is the first base on the left, second base at the top, and then the third base on the right. So U will be U, and then U. So that's phenylalanine here, for example. So UAA, UAG, and UGA, there are 3 codons that do not have tRNAs that match them. Every other one does. So we have our start codon down here, which is AUG, that encodes methionine. And we have 3 stop codons up here, which don't encode anything. So essentially, once the ribosome has one of these stop codons in the A site, nothing will come in to continue the formation of that polypeptide chain because no tRNAs come in, right? So the ribosome pauses, but it doesn't immediately dissociate. What we need is another thing. This is called the release factor. So essentially, we've got kind of few things that need to happen. One, the ribosome processes over a stop codon. So you can now see here's our that's UGA, right? One of the three. So we now have a stop codon in the A site, which you can see just here. Release factor comes in, so it basically recognizes the paused ribosome, and I don't know whether it recognizes the UGA itself. I'm not 100% sure on that. I'll look that up and educate myself. But then we have two things that have to happen. One, we've got to get this polypeptide chain off of the tRNA in the P site. So if you remember the peptidyltransferase, its job is to cut it off and put it onto a tRNA in the A site. Well, there's no tRNA there. So when this gets cleaved or released, it just floats away, right? So now we have our nascent polypeptide chain released from the ribosome. This isn't necessarily a functional protein right now. It needs to fold. It might need subsequent processing. There are a lot more steps involved in protein production than just producing the polypeptide chain. But for our purposes right now, this is the end of the translation process. And the last thing that has to happen is this whole assembly, and this is a huge complex of proteins and ribosomal RNAs, needs to dissociate. And so the energy that was used to cut off this polypeptide chain also allows the dissociation of this complex. So we now have, again, our large subunit, our small subunit, the tRNAs that have given up their amino acids, all of that kind of falls to pieces and essentially gets reused in another round of translation. So these tRNAs get recharged by amino acyl synthetase. This small subunit is going to be recruited to another 5' cap or Shine-Delgado sequence. And then this is going to, assemble over that and begin the process of translation all over again. So there's a constant cycle of stuff being used, being put back together again, being used, being put back together again, and so on. But this is why these start codons are so powerful, I guess, right? If there are any mutations which introduce a stop codon or change the frame so that a stop codon is now in frame, which is in some ways more common, that stops protein translation and synthesis dead for that gene. And typically that results in a non-functional protein. Sometimes even only a little bit can be gone and it doesn't work. One of the genes I worked on as a postdoc and actually still doing a project with my postdoc supervisor back in Penn State. It lost 7 amino acids out of, I don't even know how big it is, about 300, maybe a bit longer. Amino acids, 7, that's it, no more than that. Complete loss of function, doesn't work one little bit. You still get mRNA produced, you still get protein produced, it's just that protein doesn't do anything, it's non-functional, right? And so anytime you have one of these stop codons popping up where it shouldn't be, that almost always leads to a loss of function. Not just a little bit, but almost always entire loss of function, a null mutation. Okay. So the way that this is portrayed, right, the way that we've kind of shown it so far, seems to imply that there's, you know, a mRNA and there's a ribosome churning along it quite happily, producing protein. Well, that's a bit of a simplification because as soon as this ribosome, where's the AUG? There it is. As soon as the physical mass of this ribosome has moved away from that start codon and has cleared it, basically, another small subunit can either assemble over it if it's a prokaryote or process onto it if it's a eukaryote and start translation again, right? And this creates what's called a polysome, which you can see here. So for any given mRNA, and this also depends on the strength of the initiation of translation too, right? Because that's going to vary based on well, not necessarily 5' cap, but certainly the poly A binding proteins and other stuff, right, that kind of can interfere with translation. But say, there are no impediments to translation, you will have a series of chains of ribosomes on one single mRNA. So as soon as that ribosome has moved off of the start codon, another one can assemble. And as soon as that's moved off, another one and another one and another one. So you get these ribosomes spaced apart on the mRNA by about 80 to 85 base pairs. And so essentially what that does is it allows you to produce a lot of protein very quickly from one mRNA. So it's kind of like a scaling issue, right? If you want to change quickly as a cell or respond quickly to, a new nutrient source or new stress or being told to develop into differentiate into a different cell or whatever, right? You want to be able to do that quickly. And the whole process of gene expression is a fairly involved, lengthy, time-consuming process, right? We've kind of gone through all of it pretty much in its entirety, and it takes time to do all of this. And so as, kind of over here to the kind of left of my screen, right, we're busy transcribing mRNA, right? So we're producing mRNA species. Those are busy getting processed if they're eukaryotes. And once they're in the cytoplasm, then you're going to start producing protein, but you're going to be able to produce a lot of it fairly quickly because you're going to be able to use one mRNA template to produce multiple proteins or multiple copies of the same protein at the same time, right? Essentially what you see here. So it's not a stepwise, one mRNA, one ribosome, one protein. It's one gene, many mRNAs, hundreds of proteins via the formation of polysomes. So it's kind of like that massive expansion of going from your one bit of information to having many, 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 many copies of that protein if you need a lot at once. Obviously, there are other things you can do to kind of tamp that down a bit if you don't need that much protein being produced.
So what would stop from like making a lot of copies
Of the protein?
of like again like forming at the AUG and then starting over and over, like making more
So
copies there?
so that's essentially, good question. That's the part of the control of gene expression. And so in prokaryotes, there are structures in the mRNA which can interfere with translation if they're allowed to form. Right, so there are kind of intrinsic factors in the mRNA which can interfere with translation. We'll kind of get to how that can happen in just a little bit. In eukaryotes, essentially that goes all the way back to the 3' UTR. So there are lots of proteins and RNAs, small RNAs, that can bind to the 3' UTR and inhibit translation. And so they do that by affecting the poly-A binding proteins, right? If you remember, those are, where are we at? Right, so the poly-A tail and the poly-A binding proteins are important in the assembly of the small subunit on the 5' cap. And so if you stop this thing from bending, or you interfere with the interaction of these proteins with these proteins, that process doesn't, the initiation of translation isn't as robust. And that's before we get into the whole issue of mRNA stability. Right, so if you want to reduce the amount of gene expression, reducing the amount of mRNA that's around to be translated is one of the ways of doing that. So yeah, lots and lots of ways in which you can kind of interfere with this process, not just by how much transcripts produced, but after the mRNA is produced. Yeah, good question. We'll touch on some of those when we talk about gene expression in the next unit. Where are we at? Here we go. Yeah, in fact, it's astonishingly complex. it's the easy answer. I mean, I have a, I'm like, kind of over this Mariana Trench, the deepest point in the ocean. And, I kind of like swim along at the top. Maybe I've got a little snorkel on or something like that. And then like 11,000 meters of depth beneath me is essentially the rest of the control of gene expression, right? So I kind of like happily swim around on the very surface of that. I could not give you all of the information because that's just beyond what I know. It is astonishingly complex, particularly in eukaryotes. Prokaryotes have their pretty neat ways of doing things too. But eukaryotes are just crazy, essentially. And that's in many ways one of the answers to why we're complex multicellular organisms, is that we have many complex ways of controlling the expression of our genes. So depending on kind of, what evolution and development camp you happen to be in, and I'm a, control of gene expression developmental biologist, I guess. Essentially as we have evolved, we have gained more complex ways of controlling the expression of the genes that we have. So we may have the same number of genes as a soil nematode, but we are clearly more complex than those organisms are. And part of that reason, I think, is that we've gained the ability to control the expression of those genes in many new ways. And again, you can put, and other people argue that it's all about the sequence of the gene, right? The proteins that are produced. And you could put those two camps in a room and they'll happily like smack the monkeys out of each other, scientifically speaking, for, until the cows come home, right? So I don't think there's any easy way of like answering that argument, but control of gene expression is a big part of it. Anyway, so one of the cool things about prokaryotes, however, right? So eukaryotes is fairly straightforward, right? You produce an mRNA that gets processed in the nucleus, gets exported in the cytoplasm, and then either gets translated in the cytoplasm or translated in the rough endoplasmic reticulum, depending on where that protein is going to go. If that protein is cytosolic or nuclear, right? Because obviously you need proteins in the nucleus, that mRNA will be translated in the cytoplasm. If that protein is going to be destined for the plasma membrane or outside, say insulin, for example, right? It's I guess you could call it a hormone. It's an endocrine signal at least. That protein has to go out of the cell, right? So insulin kind of gets released by our pancreas, kind of floats around our body, and essentially tells our muscle and liver cells to take up glucose from the blood, right? And so those proteins get produced in the rough endoplasmic reticulum because they're destined for export, essentially. It's like coffee in Colombia. You keep all the good stuff in the country for the people to drink, and then you export all the crap stuff out. That's what I think anyway. Colombian coffee outside of Colombia isn't anywhere near as good. Same Brazilian coffee, actually. That's just my opinion. Anyway, but go on, Tanya, you wanted to ask something?
I just wanted to ask something. So, like the drinks, like for the gym, like the BCAAs or EAAs, when you drink those amino acids, what do they actually do? Because they're like essential amino acids and then the branched chain amino acids to
Oh,
build
okay.
muscle. What do they like? How would they work?
So branch chain amino acids, I don't actually know what those are, to be honest. Completely honest about that. But essentially amino acids is easy. Basically what it means is that we cannot synthesize all of the amino acids we need. Some organisms can. Bacteria are actually very good at that. But many animals and humans in particular cannot. So I don't know how many. I think there are maybe up to half. I have a feeling it's something like 7 or 8 for some reason. I don't know why. You'd have to look it up. But those amino acids we cannot synthesize ourselves, so we have to get them from our diet. And so the assumption in going to the gym is that you're breaking muscle fibers. That's how you increase muscle mass. It's like a repair mechanism essentially. And so by, kind of doing like lifts or running on a treadmill a whole bunch, essentially what you're doing is introducing wear and tear in your muscles. And then to fix that, those muscle cells have to make more protein to make more muscle fibers, essentially, the actomyosin, or even just for cells to kind of grow and divide potentially. And so you need a lot of amino acids to do that, obviously, if you're doing protein synthesis. And so those essential amino acids provide ones that you cannot make yourself. So if you don't have them, you can't essentially synthesize proteins. Now, whether you actually need to take them if you're exercising in the gym is another question, because if you have a decent balanced diet, you're getting all the nutrients you need. But telling people that isn't going to sell any kind of gym supplements whatsoever. So they don't.
Well, that happens with vitamins as well. You would
Yeah.
like if it's an excess, you'll pee them, so you're wasting money. But we like to waste money just to make
Yeah.
sure. I
And
do.
I mean, if you have like a fast food diet because whatever reason, then those things are really important because you won't get all of the nutrients you need. But if you have a fairly balanced diet and eat fruit and veggies and stuff like that and fish, then you'll probably get most of what you need anyway. But the branch chains, I don't actually know. I'd be curious to find out what those things actually are. Sometimes what you find with like gym and exercise stuff, it's a whole load of mumbo jumbo. It's like kind of uh, skin creams and stuff. They have nanoparticles and things in them. It's just bullshit. It's just like basically moisture that makes your skin plump. There's nothing that can get rid of wrinkles other than, well, actually Botox can, but we're not going to go there. That's something else entirely. Then you can't smile anymore. But all right, I'm going like off on a massive tangent here, and we've got to finish this stuff before the end of the class. So, oh, what are you replying to, Kim? The Botox or the branched amino acids? I don't know. I'll actually look them up after we finish class because I'm quite interested in what they are. I don't know if they're just uh where's our amino acid picture? Come on. I don't know if they're just kind of these amino acids that have like side chains and stuff like that, and so or maybe even like, bits sticking off the side. Maybe it's just another way of saying essential amino acids. Perhaps those are the ones we can't make. I don't actually know. But
Usually they have like leucine, valine, and phenylalanine. That one.
phenylalanine. Well, I
Yeah,
mean,
those three are
va
very important.
valine and leucine have, these kind of side chains kind of sticking off of them. So I guess you could call those branched. And those may also be in that group of essential amino acids that you can't make yourself. So yeah, maybe it's just saying the same thing, but in different ways. Because there are no other amino acids than we use in this, right? It's not like you can, drink some kind of funky ass ones and that will make you even fitter and stronger. It's just these are it pretty much. Anyway, so one of the really cool things about prokaryotes though is that they do couple their transcription and translation. And so if you think about a prokaryote, even though the DNA tends to be organized in one part of the bacterial cell, it's not separated from the rest of the cytoplasm by a membrane, right? So internal membranes or organelles are a eukaryotic only deal, right? You don't see them in prokaryotes. And so that means that as soon as that mRNA, and this is like what's that be? That'd be a transmission electron micrograph. As soon as that DNA starts getting transcribed, right, we get that mRNA. Once the start codon appears, a ribosome can assemble on it and begin translation. And it's really kind of neat because in a way you're cutting out another step, right? So this in some ways makes prokaryotes more efficient at the role of gene expression than eukaryotes, because as soon as that transcript gets produced, you're producing protein from it. You don't have to wait for it to be processed and then exported into the cytoplasm. It happens immediately. And this is actually a very, very important characteristic of prokaryotic gene expression, because this is one way in which they control the translation of their genes through this coupling of transcription and translation. And now, right now, that might not have any particular kind of hook or relevance to you. But when we get onto the control of gene expression, this will become really important. And you'll go back and, oh, that's what he was talking about, right? So the fact that they are coupled allows bacteria to control gene expression in a really, really interesting way. And that's kind of going back, Tanya, to what I was saying earlier, how prokaryotic mRNAs have features in them that are used to control their translation. And so it's actually it's pretty neat. And we'll get to that when we cover that in the next unit. But for now, just remember that because these two processes are not physically separated, they are able to occur at the same time. Okay, and so last couple of things I want to cover, and hopefully there'll be time for questions at the end if you have any, is essentially the genetic code, right? So like the molecular Rosetta Stone that allows that information in the DNA and then mRNA to get translated into a polypeptide sequence. And also a couple of funky characteristics called degeneracy and wobble. And so essentially, hang on, let's get down here. That's embodied by this table. And what you'll see is, right, so these are all the individual possible codons that you can have. I believe there are 64 of them. Let me just check, 16, 32, yeah, 64. 3 of those are taken up as stop codons, right? So they don't encode any amino acids, but the rest do. But we only have 20 amino acids. This is actually why our codons consist of 3 letters, not 2. If they consisted of 2, we wouldn't have enough different combinations to encode all 20 amino acids. And so, you can't have 2.5 bases, you have to have 3. And so that essentially gives us a vast excess of codons. And so there are many ways in which you can get the same amino acid in terms of the sequence of the mRNA. So for example, leucine is, actually let's use serine, which is better, that's here. You will get a serine amino acid added to a polypeptide chain anywhere where you have a UC and then a third letter. Because serine is encoded by UCU, UCC, UCA, and UCG. So essentially that means the third base does not matter. And that's what we mean by degenerate. So essentially, multiple codons can equal one amino acid. And that's what we mean by, and that's a degenerate genetic code. And so this in a way is a consequence of having 64, well, 61 really, because of these 3 stop codons. 61 different possible codons, but only 20 amino acids, right? But it has a really neat kind of evolutionary benefit in some ways, or not maybe evolutionary, that's kind of the wrong way of putting it, organismal benefit then. And what that means is that you can have, say for serine, if there is a randomly occurring mutation that occurs in the third base of that codon, so let's say in our mRNA we have UCU, and in our DNA that U gets, or will that be a T, gets changed to a C, doesn't make any difference, right? That would be a silent mutation. It would be a mutation, right? It would be a different allele of that gene, but it will not make any functional difference, because you will still get a serine produced, but it will just be encoded by potentially a different tRNA. And so you can look through here and you can see there are almost all of the amino acids. I think there are only 2. Just scanning through. Oh, just 1. Methionine, which is encoded by only 1 codon and recognized by only 1 tRNA. So if you have any mutations in this codon, no translation, because there is no other tRNA that will bring a methionine in. And this is absolutely essential for the initiation of translation in both prokaryotes and eukaryotes. And so that's what we mean by degeneracy. And this is kind of like essentially a buffer against mutation. So obviously, if you have UCU in your mRNA, you get serine. If you change one of the first 2 letters, let's say you change it to CCU, right? Well, now you get a proline, because you change it from this codon equals this amino acid to this codon that equals this amino acid, right? So that would result in a functional chain. And these 2 amino acids are quite different from memory. Can't remember exactly how. Not that much of a biochemist, right? So where that mutation occurs matters. If it occurs in the third base of that codon, most likely, not guaranteed, but most likely, that will not result in a change in the amino acid. Sometimes it will, right? So for example, if you go from a C to an A in after UU, you'll go from phenylalanine to leucine, for example, right? So that will result in a change. But by and large, it provides a buffer. And the other thing that's really neat as well is that we don't actually have 61 different tRNAs. And so for serine, I couldn't tell you for 100% confidence, but I think there's only one tRNA. That tRNA will recognize UC, and then it kind of doesn't really care about the third base. And so that's one way of kind of being a little bit more efficient, essentially, rather than having, you know, 61 different tRNAs and 61 different amino acid synthetases. We can get away with fewer tRNAs and synthetases, pardon me, because of that degeneracy at the third base in each codon. And that's called wobble. Basically, the first two bases really matter. And then the last base of the anticodon, or the 5' most one in the anticodon, essentially it's the last one in the codon itself, is much less important. And so the very last thing that I want to talk about, and this is something that I've kind of gone over multiple times, is this concept of reading frame. Right. So if you think about it, for any one direction of DNA, if we read in words of 3 letters, you can have 3 potential reading frames. So I've got to draw this because kind of talking about this stuff in words is not going to help. So if you have that sequence in mRNA, right? You could have codon 2, 3, 4. Make sense? You could also have that as your frame 2. And that could be your frame as well. There's nothing here in this particular sequence which will tell you which one. There's nothing that will tell the ribosome which one, which words are we actually using here. Is it this one and this one and this one? Or is it this one, this one, this one, and so on, right? So for any sequence of DNA and then RNA, you have 3 potential frames. And so essentially, the frame is set by the start codon. And so if you have AUG here, let me turn my laptop a little bit. That's the frame. So these, I'm not going to get rid of them because I want to take a picture. These frames, these two here, don't count. The ribosome is going to read this in these words, series of 3s. Now, if we had that instead, now the frame is going to be that. Because now this is the first word, and then each subsequent word is 3 letters after that. Does that make sense? So what would happen if we had this sequence here, right? So we're using the blue reading frame, and we get rid of that base.
It would scoot over.
Yeah, the frame will shift from this one, AUG, GUA, to this one, GAC, UA, whatever, right? That would be called a frame shift mutation, right? And so you basically go from, if you get rid of that one, you go from the blue to the red frame. The same applies if you added a base. So if you added a base or 2 bases, you would change the frame from this original blue one to one of these other reading frames, right? And frame shift mutations are really damaging. It's all right. Because almost, yeah, if I'd been clever, I would have included a stop code on out of frame in there. But almost always you will find a stop code on in one of the other frames. And so, oh, hang on, here we go. Let's kind of go back. There you go, that, haha, thinking on my feet. How about that? Mad professor skills. Okay, so in our normal frame, the blue one, these would be our words. There is no stop code on there. This was purely by accident. Now, if you remove this nucleotide, whatever that was, can't remember, and you change the frame to the red one, so now instead of being words long like so, now you read in words like so, this is a stop. And so frame shift mutations are really bad, not just because they change the frame of translation, so you end up with a protein which has nothing to do with the original gene sequence, right, and protein sequence. But you almost always introduce a stop codon. And in doing so, translation stops. So if this were a real gene, hey, you never know, it might actually be, then you would have a protein that is 1, 2, 3 amino acids long. And that would be it. This amino acid would be wrong, but it doesn't actually matter because the whole thing stopped anyway. So this is very, very different from those single point mutations that I was telling you about, which can change the codon that is present, right? So changing these letters from one letter to another may or may not, depending on where that change occurs, change the amino acid. And even if it does change the amino acid, depending on what that change is, right? If you go from I don't know, glycine to alanine, for example. Maybe that doesn't produce any difference functionally in the protein, right? So if you have come on, back, here we go, a single base change in the DNA sequence, you may still have a functional protein, right? Or you may have a protein that doesn't work quite as well, but it does have some function. Or maybe has a new function, who knows, right? Those changes, those mutations are not anywhere near as serious as a frameshift mutation. Frameshift mutations, or let's say we have a change from a C to an A, right? So instead of UGC, we now have UGA, so stop codon, right? So no longer do we have cysteine added, we have a stop codon. Oh, there you go, tryptophan's the other one with a single codon. Forgot about that one. Once you introduce a stop codon, translation stops, right? There's no getting around that. And so any mutations that introduce a stop codon, whether it's a single base change, say from, is that tyrosine? Yeah, tyrosine, U or C to an A or a G, or tryptophan or cysteine to a stop, or a frameshift mutation will stop translation and you'll have a non-functional protein, almost always. Right, so those are really serious mutations, catastrophic even.
And what happens to them? Because wouldn't they like repeat unless you have like that gene express control? What would happen like with the accumulation of just not functioning proteins?
Well, that's actually a really good question because in some diseases, that is the problem, right? So in some like Parkinson's is one of them, for example. I don't know if that's a result of a truncation or just a mutation that results in a misfolded protein. But typically you will have protein produced which is non-functional and that will be then degraded back down to its constituent amino acids. But if there is so much of that protein produced that basically what happens is you get things called aggregates, right? So that's almost analogous to prions. It's not quite the same thing. Prions are actually kind of weird and different. But if you get a whole bunch of unfolded protein or misfolded protein, and you can't get rid of it quick enough, and there's actually even a thing called the unfolded protein response, right? Which is kind of like a cellular stress response. If you can't get rid of that protein, those proteins will end up kind of like sticking together in larger and larger clumps. And that's one of the pathologies of a lot of neurodegenerative diseases, right? So ALS or Lou Gehrig syndrome, I think is caused by that in part. Alzheimer's, Parkinson's, and probably others that I don't know about are all caused by the accumulation of misfolded proteins. And so normally, this just results in a bit more work for the cell to get rid of those proteins and reuse those components, right? So it's not wasted. But sometimes if those proteins are expressed at a high enough level, or they're large enough, or they stick together quickly, or whatever, they can become a very serious problem. And that in itself will lead to pathology because then those cells essentially die because they can't clear those proteins. So that doesn't always happen with these truncated proteins. It just very much depends on the protein and what cell it's in and a whole bunch of other stuff. But yeah, there's nothing that actually stops those proteins from being produced, as far as I'm aware, in terms of the control of gene expression. Yeah, so it's pretty funky. Cool. Okay, well, that pretty much brings us to the end. I actually spent more time on that than I expected to. If you have, partly due to the cool questions, I love getting questions from my students. Makes my job a lot more interesting. If you have any questions about any of the material before the exam on Thursday, email me. and I'll be more than happy to answer them for you. If not, study hard and figure out what you don't know. That's the key thing, right? It's no good knowing what you do know. That's largely uninteresting. Figure out what you don't know, right? And that's part of the reason why I kind of ask these questions. You know, how many units of energy are used in the addition of one amino acid, for example, right? Because that makes you think about this in a different way, and then you understand it better as a result. Or you figure out that you don't know it, and then you go and learn it better, right? Yeah, the details are hard, but they're also one of the things that are potentially the easiest things to find. You know, so if I said which elongation factor is involved in bringing in tRNA, tappy tappy tap, Google EFTU, or you just look at the PowerPoint, right? So, but think about it as the bigger picture, and then those details have a way of finding their own home. It's a lot easier to remember them other than just a collection of facts. Oh, I'm glad, Leviat. It's kind of fun stuff to think about, right? Because it's just like machinery essentially. If you have a kind of a logic or mechanical based mind, this is a fun stuff to talk about because you know it's all about, you know, you need this to work, you need this to work, and then this goes from this, and ta-da, you end up getting a functional protein as a result, right? But also if you think about it of how things don't work, right? What would happen if you're in Yemen, right? And you're one of the millions of people that are currently under starvation conditions. You don't get those essential amino acids. If you don't get those essential amino acids, you can't do translation, or at least not as much of it, right? So you're not going to be able to make some of the things that are important. Your immune response suffers because you're not able to make the antibodies or whatever in response to infection, right? So think about these things in like the bigger context, and now, well, now it actually starts making a lot more sense, right?
Dr. Cook, it actually does because I did a research on Alzheimer and it was the tau protein. It's
Uh-huh.
entangled. So now it makes sense why the protein would be entangled by
Right.
like what you were just explaining of the accumulation and then producing Alzheimer. So it
Yeah,
does like
I'm not
correlate and
like
make sense.
super. I know that there's like long stuff involved, and I can kind of picture sort of how tau is involved in Alzheimer's, but I'm not well enough to explain it essentially. Um, but also because you know there are multiple steps after polypeptides are produced before you get a functional protein. So you have to take that, you know, essentially primary sequence, right, of amino acids, and you have to fold that into, you know, a complex three-dimensional shape, which like so. And so if this doesn't adopt the correct conformation, maybe because of mutation, maybe because too much protein is being produced, maybe because other things aren't working right in the cell, then now instead of taking this shape, it can take a different shape, right? And there are actually things which recognize those misfolded proteins and try and kind of like cajole them. Oh, I'm sorry, Ali. Um, I'm don't want to run over because I know you've got to get to other things, but it's just cool stuff to talk about. Um, if they can't control them into the right conformation in time, then they'll start kind of building up to the point where they become a much bigger issue. And particularly neurons are very, very sensitive to that. Other cells, like adipocytes or muscle cells, couldn't give a shit, to be honest. You know, it doesn't really matter to them, right? But neurons are very, very sensitive to that kind of issue. Cool. All right. Well, thanks very much, everybody. That was a whole bunch of fun. I really enjoyed that. I will be around in Zoom Thursday morning for at least half hour while you can start your exam. And if you have any questions about the exam during the rest of the day, just email me and I'll answer them as quickly as I can. All right, cool beans, everybody. I shall see some of you on Thursday and some of you on Wednesday. And if not, I'll see you next week.